Hi, my name is Taylor Carroll, and this is my protein project part 2. I'm going to be presenting as if I was talking to my 17-year-old sister who's in high school. Today I'm going to be talking about the melanocortin-1 receptor protein, also known as the MC1R protein. This protein is found within the cell membrane of melanocytes, which make melanin, or are the pigment-producing cells that create the color in our hair, skin, and eyes. When this protein is mutated, it causes decreased UV protection and can even lead to skin cancer. A mutation occurs in the MC1R protein due to a single nucleotide polymorphism, and this is when one nucleotide is replaced by another, therefore causing something called a missense mutation and changing the entire sequence of the amino acids. In the R67C mutation specifically, a mutation occurs when the amino acid cysteine takes the usual place of arginine. This occurs on the first intracellular loop of the protein. Normally, arginine is present, which is large, positively charged, and forms ionic bonds. But instead, here cysteine is now present, which is smaller, polar, and creates disulfide bonds. The mutation not only changes the protein's function, but also some things about the organism. In an unmutated MC1R protein, it acts as a receptor to bind to extracellular signaling molecules such as the alpha melanocyte stimulating hormone, also known as alpha MSH. This is a hormone that activates a G protein and down the line eventually tells melanocytes to produce more eumelanin, or the dark pigment within our skin. This causes the absorption of UV light and therefore protects the skin. But during the mutation, the protein does not correctly respond to the binding of alpha MSH, and therefore it throws off the whole cascade of events and less eumelanin is made. This results in a higher production of pheomelanin, which is the light pigment in our skin. This means that those with the mutation have a variant of red hair, pale skin, light eyes, freckles, and sun sensitivity. They also experience decreased UV protection and therefore are more likely to end up with skin cancer. So this is my model that I created, and here it is. The green colored pipe cleaners represent the extracellular loops, while the yellow ones represent the intracellular loops. The purple ones in the middle here represent the 7 alpha helixes that are present on the MC1R protein. And these clear pieces of pipe represent the intermembrane spaces where those alpha helixes are present. The beads that are all over here represent the amino acids that are present, and the mutation is shown over here on the first intracellular loop, where I discussed that the R67C mutation occurs, and it shows that cysteine, a blue bead, is present instead of the usual pink bead, which represents arginine. So on my representation here, the secondary structure of the protein would be represented by the 7 alpha helixes that I said were the purple pipe cleaners. And then the tertiary structure is the full 3D shape of the entire thing together. And there's actually no quaternary structure for the MC1R protein because the entire thing is made up of a single polypeptide chain. There are some limitations to my diagram, so I'd like to go over those. The first one would be that this is much larger than a real MC1R protein. Obviously, it's within the cell membrane of melanocytes, which are singular cells and are very, very, very small compared to what I have shown here. And then secondly, it does not display the accurate number of amino acids that are present. So I put quite a few beads on here representing the amino acids, but there are many, many more on a real one. And then lastly, the whole process of how the MC1R protein actively is a receptor protein is not shown because this is not really interactive and it doesn't show exactly everything that the protein does. It just kind of is like a stop motion view of it. So that's all, and thank you guys for listening.